Okay, this is the second concept in biology. So this concept deals with macromolecules. So macromolecules, there's four main ones. We'll talk about all of them. Carbohydrates, proteins, lipids, and nucleic acids. So just on the point of composition, these ones are CH and O mostly, CH and O mostly, CH, N and O, and in this one, CH, N, O, and P. That's just sort of their common compositions. There are proteins, and proteins have different interactions too. Okay, so let's talk about carbohydrates first. So this is a very sort of broad topic, a broad macromolecule. It's mostly for energy and energy storage. You have, like, for example, glucose, which is then used to turn into energy, currency as ATP. So it's glucose, which is a carbohydrate, is used to turn into, is used to create the right conditions to rehydrate ATP. So basically creates energy. So carbohydrates are for energy storage, but they're also for structure. So like the one that we talk about a lot is cellulose. So that is a polymer. So polymer, many monomers together of beta glucose. Now energy storage. Well, let's talk. This is energy itself. And then let's do one more little branch for, I don't know why I did that. My apologies. I'm going to get rid of that. Energy storage. So energy storage, like for example, we have glycogen for humans, and we have starch for animals. I mean, starch for plants, that's what I meant to say, for plants. Okay, so it fulfills a lot of functions. There's also like a general formula, CH2O to the nth. Like for example, C6H12O6, that's glucose in some form. It can be alpha or beta glucose. This is the general thing. It's like a general structure. I'll draw them very generally. It's these sort of ring structures. these little branches here, these are all carbons. There's one final carbon here. And there are hydroxyl groups placed around. But this is a alpha glucose because on this position here, so this is carbon 1, this is carbon 6, this is carbon 4, those are the most important ones probably. There's like a yeah, that's the sort of structure, and then we'll draw another glucose as a probably better to draw it like this. Just this is just for the sake of looking at the bonding. This is how we turn a monomer, so glucose is a monomer, into a polymer like starch. So starch is an alpha glucose polymer. So carbon 6, carbon 1. Yeah, so this is that, there's that very basic glucose molecule. I'll add in all the bases. I'll add in all the hydroxyl groups, just for completeness. CH2OH. This will be, apologies, OH. There's an OH up here. Okay, now so these two bonds, there's like a nice linkage that forms between these, and it's turned into a, these are monosaccharides, so that's the word we use for sugar. So 2 times, where's this E and why is it solving? I don't know. I think it's there. Okay, 2 times monosaccharide and we get a disaccharide. Okay, not too difficult, I hope. So this forms into a disaccharide. Draw this out. H2OH. So the hydroxyl here, the hydroxyl here. These structures are pretty long. Okay, so that is our molecules very generally. Nice and drawn up. You see here this bond that we form, it's like a, it's called a glycosidic bond. This one, see here, glycosidic, because we take one of the hydrogens off, or we take both the hydrogens off and we take one of the oxygens off, so we also have H2O formed, and that indicates that this reaction is condensation. Condensation, make water. Cool. So glycosidic bond, it's a 1,4 alpha glycosidic. And that indicates the carbon number, because this is carbon number 1 and this is carbon number 4. Yep, nice. The other name for this bond is, it is called it's also called a ether bond. Ether bond is a structure that looks like this. C to O is to C, like this sort of linkage. It's called a glycosidic bond. Right. Yeah. So that's basically how carbohydrates function. They form polysaccharides. So polysaccharides is like starch glycogen by just doing this times n, to the nth degree, and this is just a big chain. We'll talk about properties now of carbohydrates, because there are different types of carbohydrates. So let's properties of polysaccharides, because the polysaccharides that we're talking about, the three main ones. We're talking about cellulose, talking about glycogen, and we are talking about starch. So this exists as amylose and amylopectin. So right ahead, straight away, we're gonna draw a sort of line, because these two are much more closely related than is cellulose. Now, the similarity between all of them is that they are all polymers of glucose. So a molecule will be the same, like have the same amount of the same type of atoms, but the arrangement here is very important. So cellulose, it's beta glucose. So the difference is that it's a structure that looks like this. There is a hydroxyl group here. That's very important, and it's placed on the opposite side of the ring in alpha glucose. It looks different. This hydroxyl group is placed it's placed down here, but ignore that anyway. It's placed up here, and that's why it can form these bonds. Now noting, it's going to be like alternating. It's like an alternating shape, because this type of glycosidic bond is still going to take from this OH, this one here. So this one bonds to this one. So they're going to be like alternating, sort of looking. And that leads, cellulose is straight chained. That's also very important. Provides structure. That's very important. Like for the cell wall, for example. And it's also like it forms H bonds. So hydrogen bonding with other cellulose strands. So they like organize into like this strandy sort of thing. That's like they can like stack very easily. So they stack very easily and well. So that's what allows them to have this structure. It all ties back. And also the straight chainness allows it to have that structure. And the beta glucose also, so it really is all interlinked. With these ones it's a bit different. These are, so right here, beta glucose, and this one, sorry, wrong color, I shouldn't have switched. This one is alpha glucose, right? Okay, so that's the monomer that we saw here. These are alpha glucoses. Yeah. So with these, both of these are in some way, well, both technically are branched. There are forms of starch that are branched and forms that are less branched. Amylopectin, for example, is in starch is less branched than amylose. Yeah. Just check that really quickly. My apologies, sorry. Amylose in starch, as starch is unbranched, and amylopectin is branched. But as far as we'll make the distinction, they all link to alpha glucose. They both link to this. Now this one is humans, and this one is mostly plants. Now both are energy storage / release. Both of them use this. So this one is the differences, and I'll highlight them in a different color. So let's do this one. So this one, for example, is very, very branched. And this one is like slightly branched. In terms of density of these molecules, because this is so branched, it can like all the branching out happens that the molecules are actually like fairly like close together, and this one is only slightly branched. There's like less density overall of the molecules. So like for example, so if you look at densities, of course you're gonna have you're going to have the cellulose be the most dense, so you have these like lines, but then you have glycogen, which sort of goes all over the place and really crosses back over itself, so it's a bit more dense. That's glycogen. And then for starch, you have like more branch, but it's like more further away, so there's less branching, so it's less dense. So density, like this one is greater than this, so it's greater than this one of density. And now we can break down starch. Humans can break down starch, and we can break down glycogen, of course, but we don't have the enzyme for cellulose, so it's hard for it to break down. And also, cellulose doesn't dissolve in these molecules, like they dissolve in water, but they don't break apart. Cellulose especially doesn't break apart because those bonds are really strong. So those hydrogen bonds with itself really strong. So there's quite a big sense of cohesion with the cellulose structure. Glycogen a bit less and starch a bit less. That's why you can like dissolve starch in water. But yeah, so that's the overview of carbohydrates basically. Those three types. Yeah. Okay. So now we're going to talk about I'm going to talk about, oh, I've added like a pencil or something. Ooh, very fancy. Now we're going to talk about lipids. So lipids are like a really big class of molecules as well. To be honest, they're all very big classes. But lipids especially, that they do some things. So lipids, there's like a couple different things. There's fats, oils, and wax. And there's a lot of different functions of lipids, but one common sort of thing that you can sort of group lipids together in is that they are largely nonpolar, therefore forming hydrophobic interactions fairly easily. Yeah, so lipids as well. If we talk about something like fat, so fat is a triglyceride, very common. You have 3 fatty acids and a glycerol. So glycerol is a molecule that looks like this, basically. C. And then there are more hydrogens here. But this is also called propane 1,2,3 triol, I believe. Anyway, these fatty acids, they're basically carboxylic acids that have like a C chain that goes on forever. And again, see this part, once I finish drawing it in, I'll show. This part, these things here, they are largely nonpolar / all nonpolar. Yeah, it's like well, they're nonpolar basically. So when we join these molecules together, we get this triglyceride. So it forms. I'll omit the carbons just for the. I'll omit the hydrogens because it's easier. But it forms this ester bond here. And the carbons that just go on. Let's just see O, C, blah, 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 C, and onwards, and C, and onwards. So there's like this part of the molecule is like vaguely polar, and then there's like forces here that are polar, and then the charge is like it's not that great. There's a much bigger part of this which is nonpolar, so we consider the molecule to be nonpolar. So this is a triglyceride, or a fat. Yeah, so that's basically what a fat is. Now there are certain types of fats, like we have lipids. There's this important, it's very important in biology, this idea of a phospholipid. So it's usually a glyceride. So this sort of a structure, except on one of the carbons it's bound to a phosphate group in some way. So that there's still these fatty acids, and you essentially you have a, so phosphate is a very polar sort of thing. So you have this phosphate group, and you've joined it up to the, like the glyceride, and you have the very polar and the very non-polar. There's two components, a polar and a non-polar component. Now lipids can either be fats, oils, and wax. Wax is just like solid at room temperature. Oils are usually liquid at room temperature. Oils as well. What was I saying? So you have like an oil. A wax is basically just a it's a very long chained alcohol plus a fatty acid. Oils are just like fats, basically. Fats. Oils, the difference is pretty small. Fats are solid at room temperature, and oils are not. But yeah. So I'll write that: solid room temp and liquid room temp. Yeah. So yeah, that's most of what I can say. Lipids are used sometimes as energy reserve. So we talk about like energy density. So fats are like are twice as dense as carbs in energy. So okay, so basically for every like fat unit, it's twice as much energy as carbohydrates. There's more carbons basically, but it's just about they're more energy dense. Yeah, we use fats for insulation. That's pretty self-explanatory, I think. I mean, like whale blubber, for example. Like, heat doesn't transfer through very easily through fats. So let's talk about this phospholipid a little bit more. Basically, since there's a polar and a non-polar component, this polar component really likes going to water, and this non-polar component really hates being near water. So what happens is there's this formation of a vesicle. So when I draw like an open thing, and that's like the little tail of the I can't zoom in anymore. There's like the little tail of the thing. That's sort of the head and the tail. And in water, which is a polar substance, there's going to be this meshing. The heads go here. Forgive me for this. But they form this vesicle in water because the non-polar tails, they join together because they're more likely to form like hydrophobic interactions with each other than they are to do anything with water. So these tails, they all like form interactions with each other, and the heads all form interactions with the water. And so what you have is this vesicle formed. There are two layers of phospholipids that sort of form this vesicle, and this is basically a cell membrane. Oh, this is like a vesicle. Vesicle is a fluid-filled sac. Yeah. So there's also in triglycerides and in lipids, there's this idea of saturation. So if you have saturated, you have max hydrogens, and then unsaturated. Unsaturated means not max amount of hydrogens, therefore carbon to carbon double bond or a carbon to carbon triple bond can exist. So saturated doesn't have it, but unsaturated does, and this leads to kinks being formed. So like, observe this phospholipid. So this is a straight one, this is a straight one, and then if you have like a kinked one, see that like folds out because there's like a kink that forms because of the unsaturation. So that disrupts this sort of structure. Imagine if you have like a, let's just say there's a membrane that extends all the way. So if you have these straight, nice lines and they interact very nicely, but if you have like a kink in one, then it like pushes and it disrupts the dispersion forces and doesn't allow this nice membrane to work very well. The kink disrupts. Kinks disrupt dispersion forces. Yep. Okay, so that's lipids. Very big, pretty big topic. Yeah. Very important. It's just about this one. It's just dispersion forces. That's what holds them together mostly. It holds different like waxes and other things together is the fact that they have pretty strong dispersion forces, power and numbers basically. Yep, cool. So that's lipids, that's carbohydrates. Let's talk now about, I should draw a box around this so I don't forget where it is. Let's now talk about proteins. All right, proteins. So proteins are amino acid chains. So amino acid is a the same. So amino acid chains are like a template, basically. Proteins. So proteins are amino acid chains. So I'm gonna say AA for amino acid now, from now on, because it's just easier to write. AA. Yep, so these amino acids, they go through, you have sorry. So amino acids, let's talk about those for a second, because it's a it's vital for understanding proteins. So amino acid, basically, look, if you see this structure, this is an amino acid, C, and it joins to this NH. This is a amino acid like functional group, basically, and this forms two other amino acids through something called an amide bond, which is a bond between this group. This is a carboxyl group, and this amine group, amide. So I'll just show you what it turns into. Basically, if I add two of these things together, it looks kind of like this. So H, N, H, C, H. R here is just like a group of molecules, because this is the template for it. This R group changes, but the actual like amino part of it doesn't necessarily change. It doesn't change. So this bond stays. Now this oxygen comes out, binds to the nitrogen. Nitrogen has a little it's like a hydrogen here, or wherever you wouldn't want. Carbon, hydrogen. This is the R group. This is the nitrogen group, and we'll just put this here, because this is that amide bond that I was talking about. It looks kind of like this. They just kind of join together. Do I have this right? No, my apologies. It's wrong. Because it's hydroxide and nitrogen, so basically this entire group gets lost. So it looks like this. That's what it looks like. So the amide bond looks like this. My apologies. Just have to check these things sometimes. Yeah, and H2O. So this is another condensation. If you'd seen the other video, there's it's like the formation of oh no, We talked about this before, yeah. The formation of glucose is a condensation. So this is condensation, exactly the same. Yeah, so these amino acids, they have different R groups. So R groups, they can be like positive, negative. I mean, that really we're talking about polar and non-polar. So if you have like an R group which looks like, I don't know, like C, O, OH, this is it's acidic. It's gonna form hydrogen bonds with this thing here. But if you have something like a C and a N and a H here, so this is going to probably form like basic interactions, because it's going to like lose. So if you just look here, this one will probably lose things and become like this. And this is like a negative charge. So it's acting as a donor, so it's acidic. And this one is basic, so this will be, it'll like gain this an extra hydrogen. And this will cause it to be positive charge. Yeah, Bronsted-Lowry, nice, cool. And the non-polar groups, it's like if I just do like CH, it's non-polar. There's one more. This is fairly important for protein groups. It's cysteine. So cysteine has a cysteine, perhaps that's how you spell it. Cysteine is how you said. It has a sulfur and two cysteines. Cysteines form covalent interactions. So we talk about proteins. Protein has four distinct structures. So you have primary, secondary, tertiary, and quaternary. Okay, I'm going through these. Just remember this. These are also called peptides, and this is a polypeptide chain. Okay, very important. Polypeptide chain. So we've got this polypeptide chain, and it's got like four different structures. So primary structure is like amino acids linked. And now the forces here are covalent bonds. Now, the secondary structure of a protein is amino acids folding on themselves. So local, it's very important because it's not the whole thing, it's just a part of it. It's not the whole like protein, it's just a part of it. So amino acids folding, creating shapes. The shapes we talk about is beta plated sheets. It looks kind of like, it's like a sheet that's folded. This results from interactions between, of the protein chain just folds in on itself and it forms these sheets. Beta pleated sheets. Or we can have, then I apologize for my drawing of this, a helix, an alpha helix. And so the amino acids fold on themselves and they make like these structures, like a helix and sheets. This is local, it just exists like on a slightly zoomed out. So think of these as zooms. Most zoomed in, less zoomed in, and we'll get to very less zoomed in and less zoomed in. So these fold and they're held together by I don't know, H bonds, dipole, which I'm going to write as DDs, DD, like this. And there are some dispersion forces. Dispersions. Okay, now we're going to talk about tertiary. So this is the entire polypeptide chain, unique shape, right? So there's a unique shape that comes out of how, like, the secondary structure, like there could be one bit that's like a beta plated sheet, and then like the next part's like a helix, and the next part's like a line or something like that. And all those foldings create like a tertiary, like a 3D shape, right? And and this one as well, I suppose. So that tertiary shape, it's the entire polypeptide chain of the protein. So it's mostly in the second one, it's like the hydrogen bonds that are really doing things. Like there's little hydrogen bonds between like adjacent molecules in a alpha helix. In tertiary, there's like a big range of, because it's now like a pretty big molecule. So there's hydrogen bonds, dispersion, dipoles. There's also ionic. So if you look at these ones, you see in some reaction we could have like in certain conditions you could have ionization of these things, of these little functional groups, and you'll those charges lead to them having ionic interactions in the proteins. And also there's that covalent, so sulfate. So the cysteines, they look like, I think the R group looks something like this. So N terminates into S, and so if you have the end of one, you have the start of another, SCH2, and that sort of brings the molecule closer together, right? That's how it coils. It brings the molecule together. Yep. Cool. Cool. And that the final one is the quaternary. So this is multiple polypeptide chains chains and their folding. So this one can be like, it's all as above for forces, but basically it's how multiple polypeptide chains fold in on themselves and create this quaternary structure. This is the biggest one is the quaternary structure. It's just all the chains and they're like the way they interact and they fold in on each other. That's the sort of thing. That's what it is. Yeah. So that's proteins. Oh, and what do they do? God, it's so many things. Functions. Let's talk about function. So they build cell. So they build the cell out. They provide structure as well. The repair. Proteins are responsible for work around the cell. You can have like motor proteins that deliver certain compounds around the cell. It's very broad sort of thing. They can deal with transport too. It's a lot of stuff. It's a lot of stuff that a protein can do. Yeah, I think that's about it. So just remember, polypeptide chain, primary is just those amino acids, secondary is like a local group of amino acids that like fold in on each other or they form a helix. Tertiary is that entire chain and how that has like a very unique structure. And then quaternary, I'll draw a diagram because I think it helps illustrate the point. It's like all over the place and all these things are like going in on it themselves. It's a very complex molecule here. So we go from most basic, it's literally these atoms, and they're into a chain, and then we go to like more complex. So complexity increases. Complexity increases. Yep, nice. Okay, so we've done proteins, we've done this. This will be a long video for sure. Let's talk about, we've done lipids actually, yeah. Okay, so cool. Oh, okay, nucleic acids, finally, good. So this one will be fairly brief. I hope. So nucleic acids, let's start talking with what they sort of are there. Nucleic acids. So they deal with like for example, well, they're DNA basically. It's building block of DNA. So a nucleic acid is formed out of a phosphate plus a 5-carbon sugar. So it's either ribose or deoxyribose. So 5-carbon sugar plus a base. So base is a nitrogenous base. It's like a structure basically. They're ring structures. So you have, in terms of bases, you have A, T, C, and G. So adenine, thymine, cytosine, guanine. Oh, and DNA means deoxyribonucleic nucleic acid. Right? That makes sense? Cool. And then RNA is simply ribo. Oh, for this will be deoxyribose. Ribonucleic acid. Basically, it refers to the sugar here. Because there's 2 5-carbon sugars. It's either ribose or deoxyribose. For DNA, it's deoxyribose. For RNA, it's ribose. So these are for DNA. And then for RNA, they have all the same, except it actually looks like the same. AUCG. So U is uranine. It's a thiamine, like an analog. So it's very similar, but it's just it's what RNA uses. That's as good as I can explain that. So these come in pairs, basically. So you have, adenine. I don't know. I'll just copy over this. Hold on. Copy. Paste. Paste. No? Oh, it came up there. Paste. Paste. Okay, bring that here. So uracil is U. Uracil, right? And these are all, since they come in pairs, right? One of these is going to be a like that. One is a double ringed and non-double ringed. So if you look at the structure of adenine, it looks like it's like a ring sort of thing. This goes up. This is a sort of ringy thing. And if you have a cytosine is it's just like a normal ring, like that sort of a ring. And then you have guanine. Guanine as well is it's like a double ring thing. I'll try and draw this as best as I can. This. But you see that these ones are and thymine. And thymine will look the same as in uracil. Pretty similar anyway. So the way they link is A, T, G, and C. A to T, G to C. So you have a ring, double ring to a ring, and double ring to a ring. And this is what we'll talk about. So this nucleic acid, it's got it forms into DNA. So DNA is a molecule, right? And it's like a phosphorus. Then there's this sugar. Let me draw this properly. And this phosphorus links to another sugar further down. This links to a top bit of the sugar. Ah, sorry, there's one little thing that I'm missing here. It's an extra linkage. Yeah, that's more accurate, because there is an extra carbon there that I forgot about. There's little, I mean oxygens. So there's like an oxygen here, and there's an oxygen on top of every little molecule. So they like add. I don't want to have to cut these recordings. Bear with me. It looks sort of like this. One, two, three, four. Not exactly, but roughly. Oh my gosh. What am I trying to show here? I'm trying to show this poor molecule. So this is the 5th carbon, 5 prime, and this is the 3rd carbon. This carbon links to a phosphate. This phosphate links to another carbon, which links to this, which links to another ring. Okay, that's acceptable. You say this is carbon 5, and this is carbon 3. And they link to a ring. They link to a ring. This one little. Yeah, it works as long as it's understandable. So this structure basically is like this. The fact that there's this phosphate here and this phosphate here. It's called a sugar phosphate backbone. And that's sort of what provides structure to the provides structure to the ribonucleic acid, or the deoxyribonucleic acid. So let's say this is like, I don't know, adenine, and this one is like, oh no, this one can't be adenine. Let's say this is thymine, and this is, remember, adenine and guanine. This is a double ring. Let's say this is guanine. So this will form a little carbon little hydrogen bonds with other, like another structure that looks exactly the same, but in reverse, like upside down. Thank goodness I drew it the first time, so I don't have to redraw it. But let's just say, if I copy this, paste. Can I rotate? No, it doesn't appear I can rotate. There's a... Look, just for my laziness, search up what a DNA looks like. But it looks like this basically, and it connects downwards to. There would be another phosphate and just goes on forever. That's, I mean, there's a limited amount, but it feels like it goes on forever. And then these link to other things. 1, 2, 3, 4, 5. There's links, and then this one would be terrible looking shape. It's like the key thing to note is that one ring goes to two rings goes to one ring, and they like proceed forever. Now this molecule, let's just say there is a molecule here, would be proceeding. There'd be a 3' carbon here and a 5' carbon here in that direction, and this is a 3' carbon here to a 5' carbon here. So this one is going 3' to 5', and this one is going, looking at this orientation, 3' to 5', but looking at the orientation of this, it's running 5' to 3'. So as soon as we make our orientation, it's the one that's running. So this one is called running 5' to 3'. This is later important for respiration. But yeah, so yeah, these are the nucleotides. The nucleotides themselves, these are the bases. This forms a nucleotide. So the names of these ones are adenosine. So that's adenine. Adenosine. Thymidine. Adenosine, actually. Hold on 1 second. Well, yes, these are called nucleotides. These are the bases. They're just called nitrogenous bases. So if you go online, it's a bit strange, but they'll call it like the nucleotide itself. They have like specific names. I probably, maybe I was taught them potentially. They have different names. I'll guess. Yeah, I don't want to think about that actually. They've got some pretty terrible looking names, but the important thing to remember it's this, and this, and that makes a nucleic acid, and they're building blocks of DNA, and these things they form like this, and it's like a double helix shape. I can't draw a double helix, I'm sorry. 2 threads interlinked. No, I can't draw it. Double helix. It's like a shape. And they run anti-parallel. That's very important. So one goes 5' to 3', the other one goes in the opposite direction. Like when we're talking about carbon sugar number, carbon number in the ribose sugar. Yep. Okay. And these are, building block of DNA, genes, proteins. This RNA itself, it's the process looks like this in terms of moving from into producing protein, so producing the thing necessary to do work. So we got DNA, goes to RNA, goes to protein, right? And basically DNA transcribes RNA, translates protein. Cool. I hope it was a bit briefer. I'm sorry I had to stumble over, but the other sections were pretty solid, I hope. Yeah, cool. So this was the episode where we covered a bunch of stuff. Quite a lot of stuff, in fact. Stuff on carbs. The carbs, energy, they branch out and there are a bunch of different types. A monomer is a saccharide. Lipids, very, very broad group. You have fats and things, and mostly it's triglycerides that we deal with. So triglycerides, it's glycerol and 3 fatty acids. There are also phospholipids that they form. These vesicles, kinks disrupt those sort of interactions. But this is a membrane system, essentially. Nucleic acids, therefore, DNA. Very important, of course, very important. We did talk about amino acids, but that's a subset of proteins, and proteins is very important in the body. They've got really complex structures, and they do a lot of stuff. So yeah, hope you enjoyed. You know what I would do? I think I would put my voice on to go to sleep. Not me, myself. I hate my voice when I'm recording, but maybe that could be an interesting thing. All right, see you in the next one.